Very good morning, students. So I welcome you all to today's class, Sunday's class. Okay. So before we start the session as usual, so let us head on with our technical check. Let's see if our audio and video is clear. Okay. So I'll launch a technical check. Please let me know if my audio and video is clear. The poll should be on your screen. So give me a confirmation. If it is yes, good. If it is no, we'll fix it as soon as possible. Yes. Okay. So, all right. Thank you very much for your response. I will end the poll. Okay. So, if you have any difficulties with technicalities, just put a message or raise a bar, anything. So that will help us understand what is occurring. Okay. So today is a very interesting topic. Yesterday was a very fascinating topic. Today is an interesting topic. Why? Because in yesterday's class we spoke about what is pollination, what is the importance of pollination, what are the types of pollination and factors that help in pollination, that is pollinating agents. Today we have to take this further in understanding what happens after pollination, right? So what happens after pollination? So now what happens is when the pollen grains travel from different distances, whether it is through wind, whether it is through water, or through biotic factors like birds or insects or bees, etc. Now, once they reach their destination, what is the destination, students? It is the stigma, right? So they have to reach the stigma. Once they reach the stigma, now there is a problem. What is the problem? It is not a guarantee that every single pollen grain that is being produced will go to their exact same plants. For example, if a hibiscus pollen grain is produced in a hibiscus plant, obviously. So when it gets pollinated through insects or wind or by water, okay, usually it gets transferred by bees. But anyways, if we have so many different factors, isn't it? So when this process is accomplished, the pollination process is accomplished, it is not a guarantee that always it will land up in the same plant, same species or the same variety, right? There are chances that the wrong pollen can end up on the stigma or it is not necessary. A wrong pollen is not necessary. Now what happens? Once the pollen grain lands on the surface of the stigma, now as I told, the stigma is a very receptible organ. So since it is a very receptible organ, now what happens is it is known that the stigma will produce a certain polysaccharides and proteins which will interact with the pollen grain. Okay, and the pollen, what will happen once it absorbs these polysaccharides and proteins, now it will make this pollen grain to absorb lots and lots of amounts of water from the surface of the stigma. So now we know that within the pollen grain, right? Within the pollen grain, we had exine layer, and then we have intine layer, right? And then within that, we have vegetative cell, and then we had generative cell, okay? And it was released in the two cell state. So in 60% of the angiosperms, now these pollen grains are released in two cell stage. What is a two cell stage? One is a vegetative cell, other one is a generative cell. As simple as that, correct? Now what happens? Once they come and interact with the surface of the stigma, now as they absorb this water, now what will happen? The tube cell will start swelling. Okay. So as it swells, the tube cells, okay, will try to perforate or punch through this micropore that is present on the pollen grain. You know what is a micropore, right? The one place or the germ pore, you can tell. The one place where the exine layer is not covered. This is called as a germ pore. So as it pushes through, there is one more significant change that happens. Immediately, once it recognizes all these changes, now the generative cell will undergo one more mitotic division. We already know that this generative cell is haploid. Now, once it undergoes one more mitotic division, what will happen? Again, it will produce a nuclei, okay, which is haploid. So two nuclei are produced, which are nothing but the male gametes. Okay. So the generative cell which divides will undergo a mitotic division to produce two male gametes. Now, once this we have two male gametes and we have our vegetative cell, which is trying to push, okay, which is trying to push towards the germ pore to produce what? To produce a pollen tube. Okay, it produces a pollen tube. Now, this pollen tube will carry these two male gametes towards the, see here, once it lands on the stigma, right, the tube cell will carry these two male gametes all the way to, towards this style. See, we have this style, isn't it? This style can be of two types. Either it can be a hollow style or it can be a solid style. So what is the difference between a hollow style and a solid style, people? So if these are the two types of style, okay, one style, it can be very hollow without any tissues. Whereas another type of a style, it can be very, very solid, okay, filled with a lot of tissues. Now, based on the type of style, now the pollen tube, okay, that is being pushed can be produced two types. One type of a pollen tube, which is very, very common here in this type, it just directly grows towards the egg or towards the ovary, okay, without formation of any plugs. We call it as okay, pollen plugs. So here what will happen in the solid style, as it grows towards the ovary, now what will happen, here are the two male gametes. As the male gametes are pushed through towards the ovary, now it will form plugs. What are these plugs? These plugs are called as callose plugs. Okay, callose plugs. So these callose plugs will prevent these two male gametes from going backwards as it moves towards this solid style. Okay, it's very, very nice, right? What a mechanism. Now, these two male gametes, as it, see, it goes down and down and it finally reaches our what? Our ovary. Okay, it reaches the ovarian locule. In the ovarian locule, we already know that we have the embryo sac. So this interaction where the pollen grain comes in contact with the stigma, it interacts with the stigma, it develops the pollen tube, and then it releases its two male gametes to the pollen tube. This is called as pollen pistil interaction. It is such a dynamic interaction that it exactly knows what is self pollen and what is non-self pollen. Okay. So, chalo. So, what are these events? From the deposition of the pollen to the stigma and the development of the pollen tube and the towards the ovary. So, all this is called as pollen pistil interaction. Okay. I hope you understood. We'll discuss it when we revise again. Okay. Chalo. Now, when it comes to the deposition of the pollen grains and then controlled breeding process, we have artificially we practice two important mechanisms. These are called as artificial hybridization. So I told you that in the beginning, whenever we talk about the deposition or the pollination process of these pollens, it is not a guarantee process that the exact pollen grain will land up on the desired plant, right? So it is a highly a chance factor. Why? Because the bee do not know, okay, I have to go to visit hibiscus, next I will visit hibiscus flower only. It does not happen like that, right? So it is randomly visiting different flowers, and there it is a chance factor that it will visit the same species flowers and the pollination is accomplished accordingly. So this cannot happen when we want a desired trait. So that is why we have to do controlled pollination. To do controlled pollination, we have two important mechanisms, that is emasculation and bagging. What is emasculation? Now, let us say that we have a field, okay, we have a greenhouse or we have a farm. So in our farm, what is happening? We have we have female plants and we have male plants. We have desired male plants. Okay, and then we have female plants. Now, what will happen? There are two sets of male plants. One is a normal male plant and other one is a desired male plant. What is desired male plant? I want the pollen grains of this male plant and I want these pollen grains to go on, sit on these females. Whereas there is another group of male plants, isn't it? So what I will do is, before these male plants will mature their anthers, okay, before their anthers come to maturity, now what will I do? I will cut off these anthers of the non-desired or normal male plants. Now that will give me a very good option. What is that? Only the desired male plants will develop their anthers, okay, and then I can select the pollen grains from these anthers and then I can deposit on the females. Correct, right? So this removal of this anther or male reproductive parts, it is called as emasculation. Usually this is performed in a bisexual flower. Whereas if we talk about bagging, what is bagging? Once I deposit the pollen grains on a female plant, okay, on the stigma of a female plant, now what will happen, people? There are high chances that these pollinators keep visiting these flowers, bringing me the undesired pollen grains. Even though I cut off all the anthers, there is still the risk of these pollinators, which I cannot see. It can be a small ant or it can be a small termite. So what it will lead to? It will lead to pollen pollution or pollen contamination. So that is why what I will do is I will try to bag this pollen grain with the butter paper and then I will tie it. So once I tie this flower with the butter paper, so this is called as bagging. That will prevent the pollen pollution. Understood? So in artificial hybridization, we have two techniques. One is emasculation, other one is bagging. Okay, so let's go and see further. What happens after the pollen grain makes its entry into the embryo sac? So this is a beautiful depiction of a image or a video that is given here. So what happens usually once we have a style, once we have a style, okay, and then here what will happen? We know that in the ovarian locule, okay, we have the placenta. Attached to the placenta is the funicle, right? And then we have our ovule. So here in our ovule, we have two ends. One is the micropyle, and other one is chalaza. Okay. Now what happens is, if my pollen tube travels all the way to the stigma and then it goes and enters through the micropylar region, this is called as porogamy. What is it called as? Porogamy. Poro means micropore and the pollen tube enters through the micropore. So that is a very nice way to get the fertilization done. So this type of entry of the pollen tube into the ovule, it is called as what? Porogamy. Whereas sometimes what happens, the same pollen tube might enter. See here what happens, here it came all the way till the micropore and it made an entry. This is porogamy. What happens if the pollen tube enters through chalaza? So this is called as chalazogamy. Understood? Whereas sometimes there is misguidance where our pollen tube enters through the integuments. This is called as mesogamy. Okay, this is called as what? Mesogamy. So we have porogamy, we have chalazogamy, and we have mesogamy. Porogamy is entry to the micropore, chalazogamy is entry to the chalaza, and mesogamy is entry to the integuments. Okay, now what is the entry? The pollen tube has to entry, correct, right? Now the pollen tube travels all the way to the style, and then it is reaching the ovule, and then it has to make an entry towards the embryonic sac. Now what is happening? Through its entire journey, people, it is a long journey. Sometimes the style is so big. Now it makes us wonder, how is it possible that this pollen tube is exactly traveling in the right direction? It is quite confusing, isn't it? To help the pollen tube to reach the exact destination, Now, what is happening? You can see here the pink color one. So this is a egg cell, and the other synergid is missing. Correct, right? So there is one more synergid that is missing. So this synergid is not visible. Why? Because you can see that it starts disappearing. So this synergid which starts disappearing is called as degenerating synergid. Okay. The students, before we move further, are you able to listen to my voice? And is my teaching clear? Are you able to understand? If yes, okay, let me know. If no, also you let me know. Okay. Uh, Let me give you a poll. A, you are understanding. B, you are not understanding. Make it fast. A, you are understanding the session. B, you are not understanding the session. C, repetition is needed. It's not a technical question. It is a question based on the class. So you please let me know. Yes, students from the YouTube, if you have any troubles, you can just, okay. Hold on. 1 second. Yeah. So some of you are telling you understood, some of you are telling you didn't understand. Okay. The ones who told you didn't understand, please try to put your message, like which part you didn't understand in the chat box so that we can repeat. Okay. So let me give you an idea. What are we talking about? We are talking about fertilization process. So for the fertilization to happen, we spoke about pollen pistil interaction. That is the pollen grain landing on the stigma, how the interaction happens on the stigma, and what does it lead to. So we understood that it leads to the development of pollen tube, and this pollen tube will carry two male gametes. Correct, right? So once these two male gametes are being transported through the pollen tube, see what happens here, people. So you just look at this picture. Okay, see the pollen tube has landed on the stigma. Once it lands on the stigma, what happens? We have two male gametes, and these two male gametes travel all the way to the style, and then it reaches our embryo sac. Now, based on how it reaches the embryo sac, see, I told you it reaches the embryo sac. Okay, so here, what do we have? We have ovule, correct, right? So here we have embryo sac, here we have nucellus, and then here we have integuments, okay, outer integuments and inner integuments. And then what is happening? This is our micropyle, and this is our chalaza, and these are our integuments, correct, no people? So now we told that these integuments do not cover one region. This is called as micropyle or micropore, and this is the region where the pollen tube makes its entry. That means pollen tube carrying the two male gametes, it has to enter from here, like how it is entering in this GIF. Okay, if it enters through the micropore, I told that this type of entry is called as orogamy. If it do not enter through the micropyle, if it enters through the chalaza, that is the opposite side, this is called as chalazogamy. If it enters through the integuments, it is called as mesogamy. So these are the three types of pollen entry that happens to the embryo sac. Okay, that is all we spoke, not too much technicalities. Okay, and the next aspect what we are talking is what happens after the as the pollen tube moves towards, give me a second. Yeah, what happens, the pollen tube as it moves towards the embryo sac, see, it's a long journey. That's what I'm telling, we have a long style. So if the pollen tube has to travel all the way and it has to reach the micropyle, exactly, somebody has to guide this pollen tube. So this guidance comes from this synergid. So we know that in the egg apparatus region, we have three cells. What do we have here? We have egg cell, and then we have synergids, right? In the embryo sac, correct? So now two synergids are there. one synergid 1, synergid 2. One of the synergid, what happens? Look here, before it was present. Okay, you can see here. Before the synergid will be present. After some time, the synergid will disappear. So this disappearing synergid is called as what? Degenerating synergid. Understood? Now, why this degenerating synergy? Yeah, is significant because as it degenerates, it will produce some of the proteins and polysaccharides, okay, that act like chemical signals, okay, that will help this pollen tube to follow these chemicals or chemical trail, okay, to guide this pollen tube to come all the way, okay, and reach this degenerating synergy. Now, what happens? This pollen tube, which follows the chemical trail that is released by this degenerating synergid, okay, it makes its entry to the same synergids which is degenerating. As it enters into this degenerating synergid, it will release its two male gametes. Well, we know what is two male gametes, right? So these two male gametes were the resultant of our generative cell. Okay, so these two male gametes, once it is released into this degenerating synergid, one of them, okay, will move towards the central cell and another one will move towards the egg cell. Understood? So the one which moves towards the egg cell, now what will happen? The male gamete fuses with the egg cell to produce zygote. So this is called as syngamy. So N plus N. N is the male gamete and the other one is the egg cell. So here we'll get a 2N, that is a zygote. Understood, people? Okay, so this formation of zygote, here this will become zygote, isn't it? So this is called as syngamy. Whereas another male gamete, okay, you can see here. Wait, let me erase and make it clear for you. Okay, so another male gamete, what is happening? See, look here. Another male gamete, it will travel towards the central cell and it starts fusing with these two polar nuclei. Now, what happens? These two polar nuclei, so N plus N, one nuclei and another nuclei, they will fuse together to form what? 2N. So this diploid polar nuclei again will fuse with this haploid male gamete. So 2N plus N will give what? 3N. So that will give us triploid nuclei. So this triploid nuclei is called as PEN. What is this PEN? Primary endosperm nucleus. So what is a ploidy? Triploid. This is very must and you have to remember this. So the male gamete fuses with the two polar nuclei will give us PEN, that is primary endosperm nucleus through triple fusion. Understood? So since we have one fertilization with the egg and the second fertilization is happening with the central cell, it is called as double fertilization. And since we have three fusions, one fusion with the egg and two more fusion with the polar nuclei, that is why it is called as triple fusion. So the fertilization process in our angiosperms is double fertilization and triple fusion. Okay, you have to remember this. Understood? So you understood what is double fertilization and triple fusion. Can we go further? Yeah, did you understand? I hope so. If not, students, please be free to put in the chat box. Okay, communication is very, very important. Now, what do we have here? Here in our embryo sac, what did we have towards our chalaza? We have three cells which are called as antipodal, right? Now, our central cell has only one nuclei now, and this nuclei is triploid, which we call it as PEN or primary endosperm nucleus. Whereas we have one more cell, that is the zygote, which is fertilized, right? We have zygote and one more synergid is left out, correct? What happens to this synergid and what happens to these antipodals? Now it is very, very important that you have to understand the first thing that has to develop in this entire embryo sac is the endosperm. Why endosperm? Endosperm is a reserve food material that is very, very helpful for the nourishment of what? Our zygote. Correct, right? Even for the endosperm to develop, it requires energy. Where is this energy coming from? The energy comes from, look here, people, these antipodal cells will be degenerated and it will be supplemented as energy for the development of endosperm as well as zygote. Zygote has to develop as an embryo, right? So for that, this energy, whatever the cells that are broken down, the broken down cells energy is used for the development of embryo as well as the development of endosperm. Next one, whereas the synergy that is left out, even this is degenerated and it is used for the development of embryo as well as our endosperm. So that means in our entire embryo sac, all these three cells will degenerate, correct? All these three cells will degenerate and even the synergy will degenerate, leaving us only the central cell with our PEN and our zygote. What will happen to our PEN and our zygote? See, primary endosperm nucleus, which is triploid, it will undergo vigorous division. What kind of division it is undergoing? It is undergoing free nuclear division. Okay. What kind of free nuclear division? Free nuclear mitotic. So if it undergoes free nuclear mitotic division, what is happening? The one nuclei that was having the triploid, ploid is triploid. So now it will start dividing into mitosis, giving rise to more and more of the nucleus. Okay, more and more of the nucleus. So we have lots and lots of nucleus that is being produced within this central cell, which is nothing but endosperm. So anybody asks you what is the ploidy of endosperm, you have to tell it is triploid. Why? Because this PE and primary endosperm nucleus will undergo mitotic division to produce lots and lots of nuclei, which are again triploid. Okay, understood? So now you have to remember, people, we drink coconut water. So when we drink coconut water, it is liquid. Okay, we'll ask in Kannada, in our language, we tell neer kodi. Neer means water. So watery coconut, tender coconut, if you give, means you'll give only water sometimes. Sometimes I will ask ganji. Ganji means whenever they break the coconut. In Kannada, I'm telling, I'm translating in English. Okay. So when we break the coconut, we after drinking water, you can see that there is a very tender, the coconut pulp that we eat. Correct, right? So sometimes what will happen when you are cooking, your mother, when she breaks the coconut, there is very little amount of water and there is very large amount of solid material that is left. Correct, right? So what does this mean? So the endosperm can come of three types. One is the nucellar type, okay, or the nuclear type. So this nuclear type is very watery. Other one is haloboid type. Okay, I'll write here. Haloboid type. Haloboid means what will happen? I told half water and half semi-solid. So that is called as haloboid type. Third one is solid type, the one which we use for making chutneys or for cooking sweets or something like that. So that is called as solid type. So we have three types of endosperm that is developed. So if I ask you during the entire fertilization process and embryonic development, what is the first thing that develops? You have to tell that the first thing that develops is endosperm, not the embryo. Why endosperm is required? Endosperm is a reserve material that helps in the development of the embryo as well as for the seed formation. So that is why the endosperm develops first. Okay. So the kernel develops first so that this can again degenerate to produce energy for the developing embryo. Understood? Is it clear? Embryonic development. Can we go for embryo development? Hope so. If you have doubts, let me know. Okay. So let's go and see what is happening. Oh, sorry. Okay. Let's go and see what is happening in the embryonic development. Embryonic development is quite fascinating. Why it is fascinating? I will show you. It is an elaborate process. Okay. Now you can see here. What is happening here? You can see in our dicots, we have one cell, right? We have one cell. What is this called as? This is zygote. Okay. This is called as zygote. Now, what happens to this zygote? Basically, the zygote, once it is formed, it immediately does not divide. So it will go into a state of rest until there is a little bit development of endosperm. So once a little bit amount of endosperm is developed, now the embryonic development will take place. In embryonic development, you have to remember one important factor. So that I'm going to tell you. So what is that? Once this zygote is formed, now it will divide into two cells. So one is called as basal cell. One is called as B cell, I will write it, or basal cell. Other one is called as terminal cell. Okay, it is called as terminal cell. Now, what is happening here? The terminal cell will swell. See, it is swollen. Whereas the basal cell, it will start dividing people. How many divisions it will go? It will go up to 8 to 16 cells. It will divide to produce a long stalk-like structure. Which one? The basal cell will start dividing to produce 8 to 16 cells, which will produce a stalk-like structure. And this is called as suspensor cells. Very, very important. Sometimes they have asked in the embryonic development. What is happening here? I will tell you. So they remember the suspensor cells, which are produced by the basal cells. Now, some of the parts of the suspensor cells, again, at the end, the last cell of the suspensor cell. Now, what will happen? Again, it will divide into transverse division will happen to produce 4 cells. Again, it will produce another 4 cells. So totally we have 8 cells. Out of these 8 cells, 4 cells will produce the roots, and another 4 cells will produce the root tip and the root cap. Or you don't worry about that. Forget it. Whereas this suspensor cell is there, no, it is important. Listen. Now, you have one more cells that is called as terminal cell, right? So this terminal cell, what will happen? It will start dividing transversely and vertically to produce something called as embryonic disc, or it is also called as proembryo. So this disc-like structure is our embryo, where 4 of them will produce hypophysis, which is towards the suspensor, and 4 of them will produce our plumule and radicle. Plumule, okay. So don't worry about what is producing what. So what you have to remember, the terminal cell will produce embryonic disc and the basal cell will produce suspensor. Now, why did I tell you so many times about the suspensor is, now you know that there is a disc-like structure which is produced by the terminal cell, and you have the stalk-like structure which is called a suspensor. Now, as the suspensor grows upward, what is it doing? It is pushing this embryonic disc towards our endosperm. Okay. So it pushes deep into endosperm so that this embryo can derive more and more nutrition for its development. That is very, very, very important. So after all this development, that is the embryonic disc dividing into a disc-like structure where 4 of them towards the suspensor will produce hypophysis, and then again the 4 of them will produce your, our plumule. What is happening? We will get our globular embryo, heart-shaped embryo, horseshoe-shaped embryo, and then finally we end up with horseshoe-shaped embryo, right? So what is happening here in our typical dicot? We have certain parts. What is that? We have an embryonic axis that will divide our reserve food material into 2 cotyledons, which has a storage of food. Below the embryo axis is our epicotyle, and above the embryo axis is our hypocotyle. Okay, and then we have our plumule. Okay, and then we have our radicle. So this is what we have learned in the structure of embryonic cell. So let us see. What is happening in the dicot embryo, people? Let me check once. What is happening in our dicot embryo? First, our zygote, it divides transversely to produce 2 cells, correct? So that will divide, that will give us to basal cell, and other one it will give rise to terminal cell. Understood? Now, what is happening to this basal cell? The basal cell will undergo a lot of division to produce 6 to 10 cells. Okay, the numbers can vary, and this is called as suspensor. Okay, this is called as what? Suspensor. So in this terminal cell, what will happen? It will divide in various planes to produce what? Proembryo. Proembryo, or it is also called as embryonic disc. Understood, people? Now, the one cell, let us say that this is your suspensor, and here is our terminal cell producing our proembryo. Understood? The one cell towards our pro-embryo, it will start swelling. This is called as hypophysis, which is not necessary. Okay. Whereas, look here, the other cells in this, what will happen? There will be a division. Okay. Let me show. In suspensor cells, there will be division. So where we have 4 cells towards up and 4 cells towards down, isn't it? The 4 cells towards the pro-embryo, which are the cells? These are the cells from suspensor. Okay. The 4 cells towards the pro-embryo, okay, they will produce what? Okay, they will produce what? Hypocotyle. This is called as hypocotyle. Whereas the ones which are towards the chalaza, they will produce cotyledons. So this you forget, you remember which cell from the embryo produces suspensor, that is the basal cell. Understood? And what is the role of suspensor? Role of suspensor is to push this embryo towards our endosperm, so that our embryo will get nourishment from the endosperm. Understood, people? So what does the suspensor cells give? Suspensor cells give rise to? Our 4 cells of the suspensors will give rise to root cap and root tip. Okay, and also root cortex. The root cortex, the root cap, and the root tip are coming from the suspensor cells, that is the basal cells. Whereas the pro-embryo, we have 4 cells towards the suspensor will give us hypocotyle, and 4 cells towards the chalaza will give us plumule and cotyledons. Understood? So our embryo develops initially as globular embryo, and then it goes raised to heart-shaped embryo, and then it will give rise to horseshoe-shaped embryo. So this is the process of development of dicot embryo. Did you guys understand? If not, please let me know. Again, let me relaunch the poll. A understood, B did not understand, and C, what? Yeah, I didn't end poll. Okay, fine. Hello. And C, repeat. Okay, let me know. Make it fast, students. A, you understood. B, you did not understand. C, repeat. Student, I need your response, please. Give your response. A, you have understood. B, you did not understand. C, repeat. Simple options, okay? Whereas YouTube students, don't worry, you can just post your doubts in the chat box and we can repeat. Okay? We have a lot of strength and I need at least, what do you tell? Majority. It's a democracy. Come on, make it fast, students. So I'll end the poll. I have the majority. 2 out of 3 are saying they understood. 1 out of 3, they're telling I did not understand. Now that gives me a challenge, whether I have to repeat or if I have to move on. Okay. Color of the marker. Thank you. Yeah, sure. I hope white will work. Let's see. I'll change it. You let me know. Okay. Oh, yeah, where is that? Color. So let's go with white. Is the white visible? Is it okay? Is it fine? Or should I go with other color? Let me know. Okay. The thickness also. Fine. See, this is better, right? I don't have an option, maybe. Students, I'm not getting an option to increase the thickness. Probably I can fix it in the next class. Okay. Time constraints. Okay. Highlighter. If I use a highlighter, so would I be able to write? Okay, highlighter. No, see, this becomes this. Okay, chalo. I'll fix it in the next class. Please cope up with this class, no? Because we are running out of time too. I hope you understand. Chalo. So this is done. Development of dicot embryo and structure of dicot embryo is done. So it's very simple. Okay. So only you remember these levels. Okay. Above the level of cotyledon is what? Below the level of cotyledon is what? And what are cotyledons? Okay. What is plumule? What is radicle? Except for this, there is nothing much important. Understood? So what do we have in dicotyledonous embryo? We have an embryo axis. Okay. So that has two cotyledons. We know what are the cotyledons. Next one. Remember this. Above the axis of embryo, we have what? We have epicotyle. Okay. That will, epicotyle will terminate into plumule or stem tip. Whereas the cylindrical portion, usually the below the level of cotyledons, this is called as hypocotyle, which will terminate in the lower end. That will give rise to what? Radicle or root tip. This is very, very must. What lies below the level of cotyledon and what lies above the cotyledon, and what does it terminate to? Remember, epicotyle will terminate to plumule or stem tip. So this will give rise to your stems, understood? Whereas the hypocotyle will give rise to our leaf primordia, and then it will terminate into root or root cap or radicle. Understood, people? So the final product of our sexual reproduction in angiosperm is nothing but seed. So we have a seed, correct, right? So anyways, again, we have monocot. We know what are the monocots, right? So they do not have the division between the cotyledons, and these cotyledons are very, very negligible. Okay. So what is happening here? If you see the monocot development is quite elaborate compared to that of the dicot. So here, the single zygote will divide into 3 cells instead of 2 cells. So giving rise to one terminal cell, one middle cell, other one is basal cell. Basal cell, as usual, it will develop into a suspensor. Okay, it will develop into a suspensor. Whereas your terminal cell, again, it will bulge and it will give rise to an embryonic disc. And the middle cell will remain, just the same. On the suspensorial cell will swell, whereas the middle cell will keep on dividing to elongate the suspensor. Now, here what happens is, if you check out the process of our monocot embryonic development, so as I told you, it will give rise to 3 cells. Okay, one basal cell, and then we have one middle cell, one terminal cell. So the basal cell do not divide and it will bulge, whereas the middle cell will give rise to suspensor, and the terminal cell will grow into an embryonic disc. So here, what do we have? We have a coleorhiza, and then we have coleoptile. What is coleorhiza? So it is basically a cap-like structure, something like this, which covers the radical structures of your embryo. Other one is coleoptile. The coleoptile is another scar-like structure, which will again, or scaly structure, which will cover the plumule, okay, plumule region. So we have coleoptile, coleorhiza, and a hypocotyle, okay, in our monocot embryo. So usually you can see here, the cotyledons are reduced in the form of scutellum. Okay, so this is reduced into a scutellum. So now we have coleoptile, which will cover the shoot apex, correct? Coleoptile, which covers the shoot apex, and coleorhiza, which covers the root apex. And the cotyledons are highly reduced and they act as a scutellum. So this is about monocot embryo. So all you have to remember is the zygote divides into how many cells? 3 cells. What are those? The basal cell, the middle cell, and the terminal cell. The basal cell do not divide, it just swells. Okay. Whereas the middle cell will give rise to the suspensa, whereas the terminal cell will develop into embryonic or proembryo or embryonic disc. So this is it, people. Understood. So now coming to the development of seed. So what happens during the seed development? We understand that when we look at the when we look at the ovule, so what did we have? We have nucellus, and we have integuments, something like this, correct? Okay, don't mind the picture, accuracy of the picture for our understanding. So we have integuments and we had nucellus. So what happens is we have outer and we had inner. We have outer and we had inner integuments. Now what happens to these outer and inner integuments? Look here, the outer integument, it will develop into testa, okay, which is the outermost seed coat. And the inner integument will develop into a membranous tegument. So when we take a seed, let us take groundnut seed, which is very, very young. So when you see the outermost, it is quite hard. When you remove that, in the innermost layer, you get a sac-like structure, which looks like a membrane, right? So that is nothing but tegumen. So the integuments develop into testa and tegumen. And let's see what happens. So we know that our embryos, our ovule had micropyle. The micropyle remains open and it acts like a small pore which helps in exchange of oxygen and water, which is very, very important for the survival of the seed. Understood? Next one, if you open the groundnut seed or the peanut seed, you can see that if I divide it into two halves, at the terminal portion, you can see that there is a small scar-like structure. So the small extension or a projection or a scar-like structure is nothing but the hilum that was there from the beginning, which will help in attachment of this ovule. Understood? So these are the structures that are present in our seed, and in our monocot seed we have a membrane. Okay, basically a lot of endosperm is present in the upper region, and in the lower region we have the rest of the parts. Okay, and then we have a layer called as aleurone layer, so that will separate these two regions, which is made up of protein. So coming to our seeds, the seeds, once they are produced, they are not immediately having the capacity to germinate into a new plant. So basically they undergo a phase of rest or the phase of non-germination period. So this phase where the seeds do not have the capacity to germinate into a new plant, this is called as seed dormancy or dormancy of seed. Or it's also called as sleeping, just like hibernation. They're just sleeping, they're just preparing, and they do not divide at that stage. Once they cross this dormancy, they come to this capacity to give rise to a new plant. Now, what is seed viability? See, this is a must cycle. Dormancy is a must cycle. Seed viability is how many days can the seed be preserved or be present. Preservation is different. In a natural environment, how many days can this seed be left, have the capacity to give rise to a new sapling? So let us say we have one seed and we have the seed A and seed B. The seed A, after 1 week I start putting in the soil, it will not grow. And the seed B, after 1 year, if I put in the soil also, it will grow. So that means the seed A has low viability and the seed B has more viability. So that means how many years can this seed be potential or potent enough to give rise to a new plant? So that is called as seed viability. So we have a lot of plants. For example, we have a redwood that 10,000 years it will have the viability. We have date palm that has 2,000-year-old viability. Understood? So they have the capacity to regrow into a new plant even after so many years of not getting a proper environment. Understood? So this is about seed. Now coming to classification of seed, we know that the seeds can be divided into two significant parts. One is monocotyledonous seeds, other one is dicot seed, based on number of cotyledons. If you have endosperm, it is called as endospermic or albuminous seeds. If you do not have endosperm, it is called as non-endospermic or non-albuminous seeds. Understood? Example for endospermic is maize and non-endospermic is broad bean. Whereas when it comes to your pepper, sometimes the seed still have the nucellus. If it has a remaining nucellus, see, we had nucellus, right? Integuments, nucellus, and embryo sac. If the nucellus is remnant, so okay, so it is called as persistent nucellus, we call it as, okay. So that is called as perispermic seeds. Understood? Coming to the fruits, okay. So we know that our ovule will develop into seed and ovary will develop into fruit. Now these fruits, based on how they develop, they can be classified as follows. Now you have simple fruit. If a fruit is developed from a single ovary and a single flower, it is called a simple fruit. And they are also called as unicarpellary, multicarpellary, or syncarpous fruits. Okay. Now aggregate fruits. In aggregate fruits, we have multiple pistils. Okay, not just one ovary, we have multiple pistils from a single flower. So this is called as aggregate fruits. And then we have composite fruits. In composite fruits, what do we have? We have petals, sepals. All of them will give rise to, all of them will contribute for the formation of fruits, apart from your ovary. So when everything will participate in formation of fruit, they are called as composite fruits. So based on this, whether they are composite fruits or simple fruit, we have true fruit or false fruit. True fruit, also called a simple fruit, here only the ovary will give rise to a fruit. Understood? Example is mango and pea. Whereas false fruit or pseudocarpous fruit, here the petals, sepals, all of them will involve in the formation of, example, example, apple, pear, and then we have brinjal. So these are the examples for pseudocarpous or false fruits or aggregate fruits or composite fruits. Please remember the examples. Understood? Next one is parthenocarpy. So whenever we have an embryo development without the fertilization, this is called as parthenocarpy or parthenogenesis. So parthenocarpic fruits are very, very famous. For example, you might choose a specific variety of grapes which are seedless and tastes awesome. Okay, they are very, very sweet. So they don't have seeds, or if they have seeds, they are non-viable seeds or the seeds which do not have the capacity to give rise to young ones. Understood? So parthenocarpy is used extensively in cultivation of fruits, especially. So this can be induced by low concentration of hormones like auxins and gibberellins. Please remember this. When I produce a stress on a plant by inducing low auxin and gibberellin volumes, that will lead to parthenocarpy. Example, we have banana, pineapples, grapes. Okay, they're all parthenocarpic fruits. Okay, so we get more pulp, okay, and less seeds. Now we are also getting some of them fruits. Okay, I forgot the names. Like next time I'll try to remember what fruits we are using on a daily basis. Understood? So, and sometimes what will happen, we also use ethylene hormone and along with the abscisic acid, okay, to produce dormancy or ripening effect in our fruits. Ethylene for ripening, abscisic acid for giving rise to more shelf life for our fruits. So this is about our favorite topic is apomixis. This will show us a lot of things. What is that? In apomixis was actually developed, actually discovered by Winkler. So here, a seed develops without fertilization, and this is a remarkable process for our agriculture. Why? Because if you know farming or if you know farmers, our farmers spend thousands of rupees in buying hybrid crops for every single time they sow these plants. Okay. Now, apomixis is a process where the seeds are born without fertilization. That means the exact parental characteristics. If I have a plant, a beautiful plant, okay, so of desired characteristics, it has exceptional characteristics. So if it produces a seed without fertilization, that means I will have the exact characteristics of this plant within the seed. Correct, right? So this apomixis it mimics sexual reproduction, okay, but it is an asexual reproduction. So which has the exact rates of the parent. So this is highly helpful. Why? Because it will cut down the dependency of the farmers on these big multinational agro-based companies for hybrid crops. Okay, they can preserve the traits. Understood? So this is called as apomixis. Apomixis is formation of seeds without fertilization, and it was coined by Wickler in 1908. Understood, people? So there are several methods of the development of apomictic seeds. One is agamospermy. Agamospermy means there is no gametes involved. That means development of embryo takes place through unfertilized ovule. That means haploid gametes are not formed. Only the diploid egg, okay, the egg that is produced itself will be diploid already. So that is called as agamospermy without the involvement of gametes. Understood? Next one. Sometimes we have adventative polyembryo. So what is this adventative polyembryony? So sometimes embryo develops from a diploid cell. So basically this diploid cells is derived from the nucellar tissue or integuments. Example is citrus or Opuntia. So that will lead to more than formation of more than one embryo within one ovary. So this is called as adventative polyembryo. Next one, we have recurrent agamospermy. What is recurrent? Recurrent means repeated. Agamo means no involvement of gametes. Here what happens? There is a diploid embryo sac, that means with diploid egg, that is derived from nucellus or integument. Now what will happen? These embryo sac are diploid and they directly form they directly form the seed. So this is called as recurrent agamospermy. Understood? What is non-recurrent agamospermy? Usually a haploid female gamete, already an egg is formed, and this is in haploid nature without fertilization. That is, through parthenogenesis, it will directly form our seeds or fruit. So this is called as non-recurrent agamospermy. If people ask you what is parthenogenesis, you should tell it is non-recurrent agamospermy. Reason? It involves haploid female gametes. Understood? If they ask you what is this polyembryony? Polyembryony is nothing but adventative polyembryony, where more than one embryo forms within this embryo sac or the ovule. Understood? With the help of nucellar tissue or integuments. Example is citrus and Opuntia. And the normal polyembryony is what? Again, more than one embryo is formed in a seed. And this is discovered by Leeuwenhoek in Orange in 1729. That's why we have more locules, and then we have many seeds within same fruit, right? So that is because of polyembryony. All right, students. So I had to rush the last parts of it because of the time constraint. If you have any doubts, you can let me know in the next class. So do you have any doubts? Please let me know. You have 2 minutes. And if you need any repetition of any topics, put it in the chat box. Understood? Chalo, students. Now we can end the session. Others, if you don't have any doubts, you can leave the class. All right, students. Have a nice day. Have a nice Sunday. Bye bye. Take care. I'll see you in the next class.